As he said, my name is Chris Anzalone. I am President and CEO of Arrowhead Pharmaceuticals. We'll start with this. Some of what I'll be saying today will be forward-looking statements, and so please, if you're interested in the company and investing in the company, refer to our SEC filings. We are traded on NASDAQ under ticker symbol ARWR. We closed at 626 yesterday, giving us a market cap of just under $400 million. At the end of the calendar year, we reported about $76 million of cash. So what are we? Well, we are a targeted RNAi therapeutics company. We have assembled a comprehensive platform built around this roll-up strategy. We acquired all of Roche's RNAi business in 2011. We acquired all of Novartis's RNAi business in 2015. They had, prior to that, assembled a large variety of assets via acquiring companies, via entering into license agreements. And so what we have now beneath us is quite a large platform of technologies that enable us to do this. Well, what is this? RNAi, or RNA interference, is a way of silencing target genes. And this is a really cool idea, right? So to the extent that you know that a disease is associated with the overproduction of something, and many diseases are, we can turn that something off. And so it's extraordinarily powerful. It is highly specific. To the extent that we know what that protein is, we can likely turn it off. It is powerful in that we can silence virtually any gene, and we think we are able to develop drugs faster than a traditional a small molecule drug. And also our risk profile should be different. And here's what I mean by that. Once you validate your delivery system, once you validate that which brings in the molecules that enable this gene silencing into a certain cell type, once you do that, and once you know that it is active, and once you know that it is well tolerated in humans, you can just load that up with other molecules to silence other genes. So once you start a clinical trial for drug number 2 and 3 and 4, you have a large body of data suggesting that this could be well tolerated and this could be active. And so it's those things that make this very exciting to us. And then you monetize this whole thing by developing multiple drugs. We are not a single drug company. We are a platform company with multiple drugs in the pipeline. And so what are those drugs? Here are some of them. We have 2 drugs aimed at hepatitis B infection. We have a drug against an orphan indication called alpha-1 antitrypsin deficiency. Both of those, we have drugs in the clinic in both of those areas right now. Underneath that, we have drugs against thrombosis and angioedema. We have a drug called LPA against cardiovascular disease. Think of this as a next generation, if you will, statin almost. And then we have our first drug in cancer against a certain form of kidney cancer. I'm not going to talk about those later 3 in this talk. I don't have enough time. I'm going to really just focus on hepatitis B and alpha-1 antitrypsin deficiency. So think of us as an RNAi platform play with attractive clinical candidates. We have a large hepatitis B opportunity, and again, I'll talk about that in a minute, and novel first-to-clinic approach, and we are dosing patients in ARC-AAT, our drug against alpha-1 antitrypsin deficiency. So let's start with hepatitis B. So HBV is the world's most common serious liver infection. It's thought that 350 to 400 million people in the world have chronic infections, and this is 1 in 20 people on the planet. So this is no small unmet medical need. It's thought that about 1/3 of these patients will experience cirrhosis or liver cancer before they die, and about 80% of primary liver cancers are thought to be due to hepatitis B infection. Without appropriate monitoring or treatment, 1 in 4 people with chronic HBV will die of liver cancer or liver disease. So this really is a great unmet medical need. There is no cure right now, by the way. So many of you have heard about hepatitis C and the great strides the world has made in that disease over the last couple of years. That is now a readily curable disease. That transformed this space. We think hepatitis B is the next hepatitis C. And in fact, there are twice as many people on the planet with hepatitis B than hepatitis C. There is no cure. Unlike prior to when hepatitis C was really revolutionized by Gilead and others, we were seeing 60 to 65% cure rates. Right now, there's essentially no cure rates in hepatitis B. So it is, if we could get our hands around this virus, it would be a substantial achievement in public health, to be sure. Okay, so I'm not going to go into the science of what we're doing too much, but I want to explain the basic life cycle of the virus, just so you understand where we're coming from. So in an untreated form, like any virus, when the hepatitis B virus gets into its target cell, in this case liver cells, it hijacks the machinery of that cell to make that cell create more virus and then spits out more virus, just like any other virus. The result is that you'll have a patient will have virus in the person's bloodstream and will be contagious. That virus in bloodstream will also reinfect other liver cells, such that over time, the vast majority of liver cells will be infected by this virus. What is different about this virus is that it also hijacks the cell to make large quantities of these free proteins, if you will, free antigens. The virus only makes 5 things, and some of these are exported as free proteins, free particles. The reason that's important is because what these do is they suppress the immune system. And so over the millennia, the evolution of this virus has meant that the production of these proteins has kept the host, has kept the patient from clearing the virus, him or herself. So it's been the holy grail of hepatitis B research for quite some time to find a way to turn off those proteins. The idea is if you turn those off, then the immune system can come up naturally and clear the virus. So that's the basic life cycle in broad terms of the virus. Now, standard of care right now is a class of drugs called nukes, or nucleotide analogs. Quickly, what this does is it interferes with the replication or the production of new virus. It does a great job at that. And so if you take your pill every day, chances are you'll have very low levels of virus in your bloodstream, and therefore you won't be very contagious, and you won't reinfect your liver cells. However, you still have an increased risk of cirrhosis. You still have an increased risk of liver cancer because you're not cured. You just have lower burden of virus. Once you go off those drugs, your virus comes right back up. So it's not a long-term fix, but it does a good job right now of keeping a patient from infecting his or her partner. What's important is it does nothing for those free proteins. So a person can be on these nukes for decades, and they still produce high quantities of these free proteins. Again, the importance of that is that these are immunosuppressive, and those are still turning off the immune system as it relates to hepatitis B, so that person still can't get on top of the virus, still can't clear the virus by themselves. So nukes require lifelong therapy, and they are not curative. That is where our opportunity is, to find a curative treatment, to find a functional cure. And here's the theory behind what we're trying to do here. ARC-520, our drug against chronic hepatitis B, is intended to silence the entire hepatitis B genome, turn everything off that it makes. And again, it makes 5 things. Turn all those things off. The theory goes, if you can do that, first, you'll stop the production of new virus, and second, you will stop the production of those free proteins, and that's probably the most important thing. If you stop the production of those proteins, you can get to a functional cure. You can enable the immune system to wake up, if you will, and clear the virus. That is the theory we're going off. Okay, so does it work? So you always have to start with a safety profile. We've now been in over 150 people, and we have seen a very well tolerated drug. We've seen no signs of end organ toxicity, no discontinuations due to AEs or adverse events. The most important thing in any drug is to do no harm, and at least so far it appears that we are very well tolerated. That's good news. Now, does it do anything? Does it do what we want it to do? Well, what we learned was that it's a very complicated virus, and you can really think of this virus in 4 subgroups, if you will. Now, it's not, for this audience, it's not terribly important what those subgroups are, but I'll go over them. One of the 5 things that the virus produces is something called e-antigen, right? And some patients make e-antigen, some patients don't. And so one bifurcation of the virus is whether or not a patient makes this e-antigen or this e-protein. The second is, are they on nuke therapy? Have they been on nuke therapy long term, or have they not been on nuke therapy? So that's nuke naive versus nuke experienced. And so you have these 4 quadrants. And the reason that I bring this up is that they act very differently. These are different disease states, really, we learned. And that was new to the field. We talked about that at an analyst day we had in September of 2015, and that was really new. So we interrogated all 4 of those quadrants so far in single-dose studies. We've looked at all these kinds of patients, and what we found is that We are active in all patient types. Again, remember that there are only 5 things this virus produces, and we silence the heck out of 4 of those in all patients. Now, the 5th, so-called surface antigen, the 5th protein, we silence extremely well in 1 quadrant. We silence it in other quadrants as well, other patient groups, but in this 1 patient group, we are extremely potent. So now the question is, if that's true, and if that's enough to get to a functional cure, how big is that market? Well, about 50% of the US market resides in that subgroup. About 40% of that subgroup in Asia, in Asia, about 40% of the patients are in that subgroup, and in Europe, about 1/3 of the patients are in this subgroup. And so it's a sizable subgroup. Again, remember that we're talking about very large numbers, 350 to 400 million people in the world with chronic hepatitis B infections, and so it's a big market, I think. So how deeply do we silence these things? And the answer is extremely deeply. So I think this might be my only graph in my whole talk, so bear with me for a second. The take-home message here is that this blue line is the production of that 1 protein, of that so-called S antigen, after 1 dose of ARC-520. And this is a log scale, and so at 1.4 to 1.6 logs, we're talking about almost 99% reduction of this protein. It is gone. We are, after a single dose, we decrease production by almost 99%. Who knows what that's going to be after multiple doses, but after a single dose, it's almost 99%. Now, that's important because it could be that will get us over the hump. That if we do that, then taking away these immunosuppressive forces will enable the host's immune system to come up and clear the virus. But also, just from a broader platform standpoint, this was the highest level of gene silencing ever shown in the field in humans. It was really stunning. So I think it speaks to the platform as well as speaks to the drug. Okay, so what about the rest of the market? We talked about that 1 quadrant that I think were quite potent, but how about the other 3 quadrants? Well, we have developed a second drug called ARC-521 that is optimized to address those other quadrants. We have validated this in chimpanzees where we see better than 99% reduction of all of these proteins. We expect it to be extremely potent in these other quadrants in humans. We think that the safety is de-risked because we expect the safety profile to be quite similar to that which we've seen in ARC-520 so far, because we use the exact same delivery system, so we would expect actually quite similar results. So we go into this clinical trial, I think, knowing a heck of a lot about this drug with an awful lot of confidence that it's going to be active. Now, does activity mean cure rates? I don't know. We'll find that out. I think that we have a chance of seeing whether or not it works this year, certainly next year. But I think that's a very good chance that it will be active. It will silence all the HBV proteins, and I think we can do that in a well tolerated way. We're going to be in the clinic with this drug this summer. We've said that we're going to file an IND or equivalent by June. An IND is the document that you would file to start a clinical trial. Okay, so where are we with the hepatitis B clinical programs? This is a busy slide. The take-home message of this is that we have an awful lot of ongoing phase 2 clinical studies going on right now with ARC-520. I think that we will treat well over 200 patients this year in various subgroups with ARC-520. So can we initiate a functional cure? I don't know, but I think that we've got an awful lot of ammunition to see that, to test that this year and certainly into next year. So I think it's a very important year for us in terms of ARC-520. Now, ARC-521, as I said, will be in the clinic this year. I think it should be in the clinic this summertime because we expect to file to begin clinical trials by June of this year. Okay, that's hepatitis B. Now we'll touch on alpha-1 antitrypsin deficiency quickly before I walk away. So here's what this is. AATD, or alpha-1 antitrypsin deficiency, is a fairly large-scale orphan indication. What that means is that there's this orphan designation in the US and Europe that relates to smaller populations of patients. It's thought that there's about 100,000 patients in the US and a bit more in Europe. So here's what this is. The body normally produces this enzyme, this protein called alpha-1 antitrypsin, and one of the big things it does is it protects lungs from inflammation. So with this, your lungs are not as susceptible to environmental toxins and other things. So in this disease, alpha-1 antitrypsin deficiency, there is a gene mutation that causes a mutation of that protein. That mutation makes it hard for that protein to leave liver cells. This is all made, or most of it is made in the liver. And so what that does is two things. You have very low levels of this protein in circulation, so your lungs now are not terribly protected. But the second thing you do is you have problems within the liver, because you can imagine that the liver keeps trying to make this protein, but it can't get out. And so you can imagine it makes more and more of the protein, but that protein doesn't escape liver cells. And that causes inflammation in liver cells and then leads to liver disease. So the lung pathophysiology, the lung aspects here can be treated with enzyme replacement. There are proteins now that you can take intravenously to compensate for the low levels of protective alpha-1 antitrypsin. So that we can take care of. The liver, there is no treatment. Again, you make loads and loads of this protein. It doesn't get out of the liver. It makes those liver cells sad. So we thought it was a very straight line between turning off that gene and alleviating that liver disease. Right? So this mutant protein is clearly causing liver disease. If you just turn that off, then we think that you could A, stop the progression of the disease, and B, probably alleviate prior fibrosis and inflammation, because the liver wants to heal itself. You just take away that damaging stimulus, the liver wants to heal itself. So that's the mode of action. We're designed to stop the production of this Z protein, if you will, the mutant protein, and thereby prevent the accumulation of the disease-causing protein, allow the clearance of the accumulated protein, prevent the repeated cycles of cellular damage, and probably reverse that fibrosis. So how do you test that before you get into humans? Well, there's a mouse model that's quite good, where the human gene for the malformed protein is inserted into mice. We have tested our drug in these mice, and we have seen pretty amazing results. So shown here in the upper left quadrant is baseline at 5-week-old mouse. These are slices of liver in these mice. So at 5 weeks old, it's a clean liver. You don't see any accumulation of the protein. The protein is stained in red here. And then after 13 weeks, in the upper right and lower left, these are control animals. And so we didn't give them any drug, and you see a large accumulation of these red dots, which is the accumulated globules or protein that agglomerate together and cause stress to the liver. And then with drug in the lower right, we see none of that. It was striking. It was quite clear. We then went into monkeys, and similarly, we show that we can turn this gene off, and we can turn it off for long periods of time. If you just give doses once every 6 weeks, we can knock this down 90%, and it stays down. So we were quite happy and quite encouraged about this drug. We are now in a phase 1 clinical study in Australia and Europe. We are looking at both healthy volunteers as well as patients. This will tell us about the safety profile of the drug. We expect this to have a similar safety profile as ARC-520 because we use the same delivery system. But it will also tell us about how deeply we can knock down, how deeply we can silence this protein. With that information, we will then go into a multiple dose study, or probably a series of multiple dose studies in patients to see if we can alleviate this disease. Okay, so in closing, 2016 we think is a milestone-rich year for us. We have a whole host of things going on with ARC-520. We've got, as I mentioned, multiple phase 2 studies where we're looking at patients to see if we can get to this functional cure. I think we have a good shot because we have a well-tolerated drug and we're doing what this drug is intended to do. We are silencing the genome of this virus. We think there's a reasonably good chance that will translate into functional cures in at least some patients. So I think that we will finish enrolling a number of these studies this year. As I said, I think that we'll probably treat over 200 patients this year. ARC-521, our follow-on drug that is designed to address other patient populations, will also be in the clinic this year. We are excited about that. We will complete the alpha-1 antitrypsin deficiency studies in healthy volunteers and patients. The single-dose study will then move into a phase 2 study, and we will likely launch multiple phase 2 studies. And then finally, underneath that, our pipeline continues to expand. I didn't talk about any of this, but again, I think of us as a platform company, and we continue to move forward with several drugs in the clinic. I believe we'll be in the clinic with at least 3 of these drugs next year. So again, the way I think about us is as a platform company that is capable of developing drugs very quickly and pushing them in the clinic very quickly. And the first 2 examples of that, or first 3 examples of that, ARC-520, ARC-521, and ARC-AAT. So with that, I think I went over with time. I was hoping to take questions, but I think I'm at it. Do I have time for questions? Okay, I'm not. All right, thank you very much for your time.